All right, so once they have their kind of ballpark numbers of their count of about 100 cells or so, they're going to share that information with their lab group. All right, so we want to get at least 3 counts of mitotic cells, and then they're going to determine, of course, the average. So you can, if you wanted to, you could teach them how to do it in Excel, or they can just average it themselves. But they're going to put that 3 sets of data into a little table. So here's the kind of just a basic column table, and I kind of let them go crazy with it. At the end, of course, what they're going to do is with the average number, we ask them to make a pie chart. So it's pretty straightforward to do. So hopefully you can have them do that with their final average data. And they're going to have the different cell types labeled. So there'd be, of course, interphase, prophase, metaphase, anaphase, and telophase. All right. And then they're also going to include a picture, their picture that they took of their, if you want, you could have them, the picture of their hair. If you did epidermal cells, they could have stained it. We just want to see one picture that they did before they did the plant, if you want. And then they can actually define what the magnification level was. So, of course, the ocular lens is 10x, and then the objective lenses range. So if they were using the 4x objective lens, it'd be 10 times the 4x red objective lens, which would be 40 times the magnification. And so we just encourage them to make a little figure legend that says, the hair above shows 40-fold magnification of the hair, right? Then they're also going to be putting their picture in here that they took. And what we ask them to do is to overlay either an arrow or some sort of representation of an example of each of the of a cell type, of a cell that's actually in interphase, prophase, metaphase, anaphase, and telophase. Just one. They just need to do one good example of each one. And then in their lab report, they're going to define why that picture was, the red arrow represents a great example of interphase. The reason why I think this one is notable, or is it a great example, is because the nuclei are indistinguishable, and the cell is kind of in its resting state, etc. Right? And then they would go through and explain then the difference between an interphase cell versus a prophase cell. So just getting them writing. And they usually can cite their textbook very nicely from this. And again, if their picture stinks, if they want, I'm fine with them having Googling one of those beautiful pictures of an onion root tip and using that as their example to overlay the colors or the arrows or the letters or whatever it is that you're going to have them identify their different cell types or their different mitotic stages at. Hopefully that makes sense. I'm also going, and then there's some questions at the bottom which are very straightforward. It's almost embarrassingly straightforward. And then they'll put their references in at the bottom. All right, so it's usually a very easy, straightforward lab. I actually do have a recording of when we had to do the online version last year. We actually had to quarantine for a period of time. So I do have a recording of this lab that was remote, and it should also be in your teacher materials that are in the lecture. Oh, did it get dis- I actually have a copy of it. I saved a copy, so I'll find that and I'll make sure that gets uploaded to the YouTube videos. Okay, so that might be actually in the YouTube. If you need any data, there is a little spreadsheet here that has some of the data I got from 1 year. And again, if all goes to hell, right, you're going to see, of course, that most of the cells are going to be an interphase, right? That tends to be what we see more often because more cells are kind of resting. All right. So hopefully that helps. Have fun with this lab. I think they typically like it a lot. All right, good luck.